So I have a PhD in basic neuroscience, and I work over here at Stanford with Dr. Axel Wunder. He's a pretty big guy. But anyway, so I'm gonna just like go over really basically what our lab does. So do you guys know, I mean, you obviously know some stuff about brains, I guess. Do you know anything about synaptic vesicles? Like, is that a thing? Okay, you guys knew. Man, my day in high school, we didn't know what a brain was. All right. So, yeah, let's go over some just basic numbers in neuroscience. So your brain has about 86 billion neurons. That's more than there are stars in the Milky Way. And that's also slightly more than the number of Homo sapiens sapiens that have ever walked on this planet. It also, you could throw in Homo denizen and Homo erectus probably, and you still wouldn't get to the number of neurons that you have. On top of that, each one of your neurons can make up to 1,000 connections. Come on in, there's pizza. No teacher. So each one of these neurons can make a connection with another neuron and make up to 1,000 of these connections. They're called synapses, which means that the number of connections you have in your brain is 1,000 Milky Ways, or 86 trillion connections. And at each one of these connections, you have synaptic vesicles, which you guys know about. They package neurotransmitter. Maybe you guys know about how they actually release neurotransmitter. But basically, they're very, very important, right, for the total function of your neuron. So we have this model of how synaptic vesicles really work. The major point being that you have vesicles full of neurotransmitter. They come, they interact with the plasma membrane of the presynaptic terminal. All of this machinery down here does some stuff, and then you get fusion, and you release neurotransmitter, and the neurons are able to talk to one another chemically. So our lab is interested in how exactly this step works. And one of the problems with this model is that realistically, neurons are actually crazy complicated. They have thousands of different proteins in here. They have, like I said, hundreds to hundreds of thousands of synaptic vesicles. So how does the neuron really organize all of this stuff? And then on top of that, synaptic vesicles are themselves really complicated. They don't just have two proteins sticking off of them. They're full of neurotransmitter. They actually have tons of different proteins on them that each play certain roles in trafficking, locating them to the right space, and then actually being used in fusion. So what our lab is really interested in, these are just some minimum requirements that you need. What we're really interested in are these synaptic repins, which are the proteins that are required to actually cause all of this fusion. In addition, synaptic tagline, which you guys also probably know about. And for my projects, these are kind of important, but we're just going to forget about that for right now. So what do I actually do? There's this recent theory that these synaptic vesicles are actually very different, even within a single neuron. They're not all the same, and that these differences actually have very severe impacts on communication, and that's really important. So what I do is I actually take a bunch of animals, and I take their brains out, and I grind them up, and then I purify these synaptic vesicles out of that. Then I look at what proteins are on them using a technique that, frankly, I don't understand either, so it's all good, called mass spec, so don't ask me about it. Then I want to do some other stuff. We also want to see whether or not these vesicles actually have specific locations in space that they occupy. So we use this really cool super resolution technology called STORM, stands for Stochastic Optical Reconstruction Microscopy, which is just really fancy, yet did not win a Nobel Prize. She actually got really screwed out of that. So STORM basically lets you beat the light diffraction limit of in microscopy. And then we want to see whether or not these putatively different synaptic vesicles actually function differently. So my lab, one of the big things we did was create this totally synthetic system. So we can reproduce neurotransmission in a test tube, essentially. And what I was brought in to do was to add one more step of complexity, like make it a little bit more real by using the synaptic vesicles that I purify in this assay. Okay? So to do that, I use these techniques. Protein organelle purification, STORM, we use X-ray crystallography, I do tissue culture, I do electron microscopy, we have this fusion assay, I collaborate and do mass spec with people at UCSF in Boulder, Colorado, and animal behavior and electrophysiology. The stuff with the checks are the things I actually understand. The things with the question mark is the stuff that I don't really know. So when you get to be a PhD, you don't have to know everything. You're not expected to know everything. You can still do your job, and it's fun. So, like I said, I grind up animals, grind up their brains specifically. Then I put on an antibody. I do an immunoprecipitation. I don't know if you guys know what that means, but basically I put an antibody that recognizes a vesicle protein. Then I put on some beads that recognize that antibody. Then I can wash everything else away, and all of my vesicles are stuck with beads. So here's an electron micrograph of these synaptic vesicles. They're pretty pure, so these little guys are synaptic vesicles. You can see there's still some like garbage and junk in there. To give you an idea of the size of this, from my finger to the end of the light over there would be the diameter of one of your hairs. So these are pretty small. They're tiny little things, and that's made them very hard to purify and isolate. There's only 2 labs in the world that do it, and we're one of them. The other one's in Germany. So there's a little close-up. You might be able to see, probably not from where you're all sitting, these little black dots. Those are gold particles that are towards other synaptic vesicle proteins. So anyway, this was a movie that's not going to work, unfortunately, that has a brain, and I cut it up. So just imagine a little mouse brain, I cut it up. This is what neurons look like when I culture them, so we're moving on to Storm right now. So with conventional microscopy techniques, you have these antibodies, they have dyes on them that release light when you shine a certain wavelength of light on them. With conventional microscopy, if you had 5 of these very close together, you couldn't tell them apart. It would just appear as one dot, and so this is kind of the best you can get, which is like super pixelated and really terrible. With Storm, you actually use dyes that are not what we would typically consider good. So you shine a light on them and they blink randomly. And then you take these really long movies, you average all the blinks together, and using math, you can actually figure out where the exact center was. And so now you can resolve these, what was really crappy here, you can now see these like very beautiful strands and everything. So that's one of the other techniques I use. Then we have this fusion assay, and well, I'll tell you about the fusion assay and then I'll show you. So our fusion assay, what we did is we made these synthetic vesicles using actually pork brain lipid extract. So somebody else, I guess they take a bunch of pigs and they grind up their brains and then they solubilize all the lipids and then they sell that to you for a ridiculous amount of money. So what we do is we purify, we express these proteins, all of the brain proteins in bacteria actually, and then we purify them out of bacteria and we can put them into these lipid vesicles and we can actually trigger fusion, just like you would in your neuron, except this is totally fake and in a test tube. So what I do is I put on real vesicles, they kind of look like this, and when they fuse, we use this dye that is very special and it self-quenches, so in small volumes it's off, in large volumes it gets brighter. So you can actually see like fusion events happen, and it's really pretty, and that's my data. So let me show you briefly a video. So in order to do that, I had to build this setup. So let's see if this works. So this is what one of these rigs looks like. And this is something you would do. I've never built one of these before. It's not something that I had from memory. It's something that you just kind of like go through. And that's one of the major things you need when you're doing a PhD is creativity and the ability to just figure stuff out. You're not going to be memorizing tons of stuff. You just need to think about what you need to do and then you do it. And this thing is probably, all that stuff on there probably costs towards $500,000. So you get money from grants, your boss, when you're a postdoc like me, I don't make that kind of money. So my boss buys all this stuff and then we assemble it in order to get what we want. So what's happening, these are red, green, and blue lasers. We bounce these lasers off of these mirrors. So the red one's going over here. It goes through, this is called the dichroic mirror that reflects certain wavelengths of light at 45 degrees but lets others pass through. So the red light can actually go straight through. Then it's gonna come over here and it goes up here eventually. The green light kind of takes a similar path but bounces off of this mirror, goes down, and the blue one also. I also built this rig so that the blue light can actually do 2 kinds of fluorescence. So it's just like increasing the utility for, we don't need that, but it might be useful in the future, so I did it. So yeah, now I'm building the emission pathway, so that's the camera. It's like really fancy and expensive. And there I'm trying to figure out the software, it's terrible. And there's like the laser lights. So this is just something that had to be done, so I did it. It's not like this was part of a proposed project or anything, it was just on the way. And that's a lot of what science is, you're just kind of winging it. You don't really have too much of a plan. Okay, so there's that, and I'm just trying to align this thing. So yeah, but it works. This isn't data from that, but that one does work. So the last thing we do is X-ray crystallography. And this stuff it's very, very powerful, but it has some pretty serious caveats. So what you do is you purify your proteins, you mix them together. If you're really, really, really lucky, they grow crystals. These are actual crystals that we've grown. Then we took it over to SLAC, which you guys have probably driven over. That's 280, goes across it, I think. So that's a large particle accelerator. You slam photons into this, into your crystals at very high speed, and you diffract out images. Maybe you guys already know this, but those are like actual atoms in space, and then you can do some fancy programming, and you can get an actual structure. So you can build this, and these are where those, every individual atom is actually located thanks to these images. Now you can take this, and you can go in here and say, like, here's an interaction surface. I want to change some of these amino acids and see if I can disrupt that interaction surface. That's what crystallography lets you do. So we can do that, then we make a virus that will express that mutated protein, we stick it in an animal, and we give it to another lab to tell us what gets screwed up and what's okay. So our lab, we really focus on a very, very, very small aspect of neuroscience. But then that tiny thing is very important for the function of these organelles and release a neurotransmitter, which in turn affects how neurons work individually and thus in a circuit, and then how parts of your brain actually work together to ultimately make up who you are and everything that you can be. So that's the science portion. So the rest of this, I'm just going to talk about how I got to where I am today. So I'm from Golden, Colorado. It's the home of Coors. I say this because there's no teacher. Don't ever drink that beer. It's terrible. We, as kids, routinely urinate in the river that they use. So don't go there. I mean, go to Golden. It's beautiful, but don't drink Coors. So when I left high school, I really, really wanted to either be a film director or an MD PhD. Those were like, that's what I was going to do. And then my second choice was a scientist in like plant physiology or population biology. Then I went to Reed College, and you guys probably can't read that, but there I discovered that being a film director is not a super great job. You're really poor for a really, really long time. And I found out that being an MD PhD, you have to have like memory, which I don't have. So then I was like, okay, maybe I could handle being a doctor, I'm not sure, or I could do this PhD in either plant physiology or neuroscience. And I did my undergraduate thesis. That's one of the things I had to actually do at my specific college. You guys might not have to do that if you go to a big university. He was a neuroscience guy, and he was awesome. So then I decided to be a lab tech for 1 year, which one of your former graduates is currently a lab tech for us right now. And that I highly recommend. Like, take a year off. Your parents might be mad, but just do it. You'll get paid. And see whether or not you're going to be able to handle doing a PhD, or if you're going to handle the process of science really well. I ended up loving it, and I had a really, really awesome PhD mentor that I teched with down in Dallas. So now I am pretty certain that I'm happy where I am. But just keep an open mind. Like, you guys are in high school. You have plenty of time to figure stuff out. And also, what you do your major in up here really does not matter for what you end up doing later on in graduate school. As long as you're smart, people will know it. So, all right, college versus university. I went to college, so I'm super biased. They're way better. Smaller class size, which is great. You get a more rounded education if you go to a liberal arts college, which I also really like. But you don't really have like the major options and the course options that you would have at a university. So like Reed College doesn't have a neuroscience major, but you don't really need one. I was biochemistry and molecular biology. My girlfriend was biochemistry and molecular biology. I work at Stanford. She works at Happy Hollow Zoo playing with animals, and she gets paid for that. So, you know, I mean, your major doesn't really determine what you're going to do with the rest of your life. There are some downsides to being in college versus a university. You don't have as many resources, which is kind of good and bad. It makes you improvise and stuff. So I used duct tape a lot when I was doing my undergraduate thesis. Duct tape lots of stuff together. They're obviously probably more expensive, especially for you guys. For me, coming from Colorado to come here from out of state would have been more pricey than for me to go to a private college, because you guys are crazy. Universities, it's also really hard to stand out, which I think is actually a pretty major problem. And the other part that I didn't put on here is a thesis. Theses, do one. If you have the option to do a thesis in college or university, and if you do, you'll be so much better prepared for graduate school than people who don't. Okay, so then I drew out like a little, your guys' future, which is probably not going to be accurate by the time you guys are there. So this is, over there on the right is the actual year you guys would be doing this if you're a senior now and graduating. So here's like timeline by 2 years. Graduate school is really nice. This is also to illustrate the only reason you should do an MD PhD, and there literally is only one reason to do it. So in graduate school, you get a stipend. You actually get paid to do science and like goof around. So it's roughly 30,000 right now. By the time you guys do that, it'll be a lot more. Then after, so the average is like 6 years. Then you can be a postdoc. All of these arrows are postdoc. They're required for everything. Industry now requires you to do at least 2 years of postdoc before you jump into the workforce. This is the average amount of money you'll get paid. Every postdoc gets paid a certain amount, so I'm like at 57, and you are guaranteed a pay raise every year, which is also really nice. And you get free healthcare, which is awesome. And then you can kind of go into tenure track research, or you can just, at any point, you can kind of hop off and go back into industry, and they'll pay you just a silly amount of money. For medical school, the average price is about $50K, and right now the average medical student accrues about $250,000 in debt just from medical school. So that part's pretty rough, but then you make money like crazy, which you don't make as a PhD. So residencies vary depending on what you want to go into. If you want to do family practice or pediatrics, it's 3 years. If you want to do like surgery, where you make the big bucks, it's 6 years, I think, and some of them are 7. I think for like plastic surgery is like 8, something crazy, but you make like just an obscene amount of money. So the real reason to do an MD PhD, so MD PhDs, you have to do both, and they set you back 2 years, and that is by far the worst part, because you don't get anything out of it at the end. They're not going to pay you more because you have 2 degrees. You're not going to be picked over other people. Well, you might be. If you get an MD PhD from like university of nobody cares, they will hire an MD from Harvard over you any day. It doesn't matter that you have both degrees, they don't care. So if you want to do MD, just do the MD. If you can't afford to do MD, then do the MD PhD, because there you get a stipend. So you will get paid more than a graduate student to do your MD PhD program. That is literally the only reason to do it. There is no other reason to do an MD PhD. Nowadays, like the idea of the crazy scientist that like experiments on his patients, that doesn't exist anymore, because you don't have time. As a scientist, you have to write grants. You don't have time for clinic. As a medical doctor, you have to be in the clinic. You don't have time to mess around with experiments. So like, you're not going to be like Sean Connery running around in the Amazon finding like ants that cure cancer and grinding them up and like figuring out what it is and giving it to people. You're going to have collaborators that do that. So you'll collaborate with Sean Connery and you'll give the stuff, or you'll be Sean Connery and then you'll just give the stuff away. So like, that's it. So what does my like actual day-to-day look like? You can see like some of the experiments. Here's what we use to purify protein on these columns. This is the FSL stuff over at Slack, and this is my rig that I'm so proud of. So I roll in, like, this is the greatest part of that PhD. So I roll in whenever I want, which is typically 8 o'clock, because then I feel like a bum. I don't. I can leave whenever I want, which sometimes is like noon, but other times it's like 4 a.m. And I've spent 36 hours in the lab before, but it's because I wanted to. It's not because anyone made me. So that's the nicest part. Also, you get to do what you actually want to do. You get to design your own project. You get to do the experiments for your project. You get this total freedom that is really awesome. So, for example, September, I only worked 3 days. The rest of the time, I was on vacation. You know, but then in July, I worked every single day of the week. So you can kind of do stuff whenever you want. We don't have sick days. We don't have a number of days you get off, and it's all salaried, so it's not per hour. So you kind of just do stuff. So like, yeah, here I went to Wyoming. These are all trips I did this year. So this is Colorado River. This is canyoneering in Utah. This is the Wind Rivers Range in Wyoming and / Montana. And I was hiking with my dad and my dog in Colorado. So there are also other things that we desperately need smart people to go into that are not MD, PhD stuff. And one of those is communication and science outreach. Like, it's absurd that creationism is still a thing. That is crazy sauce. We're a developed nation. I mean, that's nuts. So we need like really good communicators. We need really good science outreach. We need good people in museums. We need people that are changing museums to actually keep up with what is relevant in science. Like, we don't care about dinosaurs anymore. We need molecular biology stuff, right? Dinosaurs are cool, but yeah, we need molecular biology stuff. We need kids to be interested in this. Like, you guys at least get coding. Our computers were terrible when I was a kid. There's industry, which I didn't talk about too much. Industry is a lot like getting your PhD. It's a lot like that, except you don't have creative control over what your project is, so you're kind of more or less told what to do, but it is nice that you have a regular schedule and you're paid really well. So if that's something that you want, if you want a 9 to 5, then by all means do industry. If you want like a crazy schedule that's hard to predict, then stay in academia. We also desperately need people in law, and so like it's crazy that it's easier for me to get cocaine to test on rats than it is for THC, which is insane because we're voting on whether or not to legalize marijuana, right? And I can't do research on it. Nobody can, but I can tell you that coke's real bad. So, like, we need people changing laws. We need people working for funding sources, because it doesn't matter how good I am or what I produce if the people that fund me drop out. So we need people that do that, go around and collect money for us, that work for big foundations. And we also need people in administration, like hospital administrators are terrible. We need better hospital administrators. Academic administrators are either really good or the worst human beings on the planet. So it sucks because they actually make your world go round. So they can either really help you out or just hamstring you like crazy. So that's something to keep in mind. So that's it. That was like blasting through it. But yeah, so if you guys have any questions about stuff, and you can also even give them my Stanford email. So anything about the science or the your future?
Yeah. When did you know you wanted to go into science? Like, what was it about research that got you hooked?
It was the kind of the freedom that you really get, where you can do whatever you want. You can roll in whenever you want, leave whenever you want. That really got me. That was part of it. The other part is that I get really excited about. So when I wanted to be a doctor back in the day, I really wanted to help people. And I had this epiphany probably in graduate school where, yeah, being an MD, you can help one person, you can fix their problems. The thing that would drive me crazy is that the next week you would see someone with literally the same problem. Right? You're not actually curing a disease. You're helping people, yeah, for sure, definitely. But it would drive me crazy to not be able to actually stop the thing that is doing this. And so that's why I wanted to go into PhD. And then in doing that, I realized that we actually know very little about how neurons work. I mean, neuroscience is an incredibly new field. By the time you guys graduate college, pretty much every school will have a neuroscience major. I mean, more do now, but when I did, there was no neuroscience major. It's going to be its own separate thing. There'll be biology, chemistry, physics, neuroscience. It'll be its own.
Do you think it's going like a lot right now?
Yes, it's huge, because everyone is like, okay, my parents are baby boomers, so they're all gonna go crazy really soon, and there are more of them than there are of my generation. So we're really worried about it. And there's you guys probably heard that we're really close to figuring out Alzheimer's. That's familial Alzheimer's. That's 1% of Alzheimer's. We don't even know what causes the 99% of Alzheimer's. So with just random acquired Alzheimer's, like, how are you gonna figure that out? especially if you don't know how a normal neuron works. So that's why that was the thing that snapped in my head. But by all means, do an MD. don't just listen to a jaded old guy. Yeah.
With regard to like the technical stuff for like the assay, do you do it in like plates, like 96 well stuff? Like
No,
that?
it's actually, we make these little channels with glass, so it's just microscope slides,
Oh, cool.
really. And then yeah, we have this way to attach all the vesicles there, and then we can do 5 at a time, like 5 like channels. And then we've got a kid working on a new technique that will let us look multiple times within one channel instead of just like blowing the whole thing out.
Where do you get the brains? Like the animal brains?
The animals, so you get them from Jackson Labs. So we don't have an animal colony, and we're too cheap to get one. So I actually just use Tom Sudoff's animals. He won the Nobel like 2 years ago, so he's at Stanford. So we just take his leftovers.
Does he have an animal farm?
Yeah, kind of.
Really?
I mean, it's called a colony. So underneath Stanford, I don't know, none of you guys are like PETA people, right? Like, you have to tell me if you are. So under Stanford, there's this huge mouse colony and rat colony. So there are like thousands of mice and rats under there. And they're genetic knockouts, they're genetically mutated, and they're whatever you want, pretty much. And so you order those from a place, Jackson Laboratory, I think it's in Maryland. And they ship you whatever mouse you want for a lot of money. Yeah.
Oh.
So that's where we get the mice. You don't like run outside and catch them. Yeah, when we say wild type, we don't mean wild. They're actually very inbred. Like all the rats go blind after like a year.
Oh,
Yeah, they're super inbred.
that's terrible.
Yeah, that's the pain. So actually, one of the advantages of this system is that my goal by the time I leave is to actually use people. I mean, people that died naturally. I'm not going to go out and like whack somebody. But yeah, I mean, in theory, the advantage of this whole synthetic system is that I could ground up human brain, and then I could use that, which so far no one's been able to use any human tissue in a functional assay like this.
Why is that?
Because when you dead, your neurons really stop working. But this bypasses that, right? Like I don't have to worry about the function of the cell. I can just purify out the little organelles and then use them outside. So we skip that whole thing. So that would actually be